Let's see the introduction of operation of micropipettes. Now micropipettes are utilized in the laboratory to transfer small quantities of liquid, usually down to zero point 0.1 microliter. They are most commonly used in chemistry, biology, forensic, pharmaceutical and drug discovery labs among others. Now these are some common sizes and common names of micropipettes. See, the first pipette is the P2 pipette. The volume range is 0.2 to 2 microliter. P10 micropipette size, that is 1 to 10 microliter. P20 2 to 20 microliter. P100 20 to 100 microliters. P200 20 to 200 microliters, and P1000 100 to 1000 microliters. So these are the some micropipette names and these are the volume ranges. Now let's see the structure of micropipette. Now see, at the top the micropipette contains piston button, then there is ejector, and this is the big digital window where we can see the volume arrangement, and this is the nozzle collection, and this is the original suction head. Now if you wanna take the sample, then we have to take with the piston button, and if you want to eject the tip, then we have to eject the tip with the help of ejector. After selecting the volume, we can see the volume in the digital window, and these are some parts. Now let's see stepwise operation of the micropipette. Now first one, we have to set the volume, then second, attach the disposable tip, then depress the plunger to the first stop, then take sample, then draw up the sample, then take pause, wait a few seconds to ensure that the full volume of sample is drawn into plastic tip or not. After that, we draw a tip. Now here we have to remove the tip from the sample liquid. No liquid should remain in the outside of the tip, and if liquid is there outside the tip, we have to clean with the help of paper. Then dispense the sample where we want to dispense. Now here how to dispense it: touch the tip at the end of the side wall of the receiving vessel, depress the plunger to the first stop, pause it, press the plunger to the second stop to expel the initial liquid in the tip. Then withdraw the pipette. Now here, after the dispenser sample, withdraw the pipette with the help of plunger, fully depress the withdraw the pipette from the receiving vessel carefully, sliding the tip along the wall of the vessel. Release the plunger. Now here, gently allow the plunger to return to the up position. Do not allow to spring back. Last one, that is we have to discard the tip. Now here, discard the tip by depressing the tip ejector button as shown in the picture. A fresh tip should be used for each sample to prevent sample carryover. Now this is the picture where the ejection is happening.